Hey everyone, welcome back to our Bioinformatics series. So far, we have learned to represent, manipulate, and read DNA sequences from files. We have even simulated transcription, the process of creating RNA from a DNA template. So today, we will try to finish that part. So we are going to explore the central dogma's final step, which is translation. This is where the cell reads the RNA blueprint and builds a functional protein. So we are going to teach our computer how to read the genetic code, just like a ribosome does in a cell. So let's start. Right. So how does an RNA sequence made of A, U, C, and G gets turned into a protein made of amino acids? So that secret lies in the genetic code. The cell reads the RNA sequences in groups of three letters called codons. Each codon, like AUG or GCA, corresponds to a specific amino acid. Think of it like a dictionary where the words are codons and definitions are amino acids. So there are also special stop codons that tells the cell when the protein is finished. So in Python, the best way to represent this dictionary is, well, obviously a dictionary. So I prepared one here. You can later on copy this directly from the notebook once it is uploaded. So in a new cell of our notebook, let's define the genetic code. Right. Now, so here the three-letter codons are the keys, and the like these, and the single-letter amino acid codes are the values. I have also used an underscore here to represent the stop codons. Right. So now that we have our dictionary, let's build a function that can translate an RNA sequence. The function will need to read the sequence codon by codon, look up the amino acid, and build the protein sequence. Right. So let's start. RNA sequence. We will define protein as a blank for now. Then we will run a loop for I in range 0, length of RNA sequence minus length of RNA sequence divided by mod 3, and then okay, inside that we will do RNA sequence from I to I plus 3. Okay, then we will define the amino acid as gen code codon, and then if we hit a, we need to close this. Then if we hit an stop codon, if amino acid is equal to underscore, then we simply stop the loop. Right. Then protein is equal to amino acid, and then we return protein. Right. So let's break this down. Our function translate takes no RNA sequence as input and starts with an empty protein string like this. Okay. So the for loop is a little tricky. It iterates from the start of the sequence to the end, but in steps of 3. Right. The length RNA sequence mod 3 part clearly handles the sequence lengths whose handle sequence whose length isn't a perfect multiple of a 3 by simply ignoring the 1 or 2 leftover bases at the end. And inside the loop, we grab a 3 base slice, our codon, and we look up that codon in our GenCode dictionary to get the amino acid. Crucially, we check if the amino acid is a stop signal. Okay, so if it is, we break out of the loop immediately. If it is not a stop codon, we append the amino acid to our protein string, and finally we return the complete protein. Okay, now for the full simulation, let's put everything we have learned together, and we'll start with a DNA sequence, right? So for that, let's use a real world example. I have taken the start of a human insulin gene, which is like this much. Okay. And after that, let's start with the transcribing. Right. So RNA sequence is equal to DNA sequence dot replace T with U, and then print RNA sequence like this. And then we translate the RNA to protein is equal to translate, and then we enter RNA sequence. Then we print it, protein sequence is protein sequence. And when we run this, okay, we didn't run this code. Okay, we, now after running the above cell, we will run this again, and here we have it. Perfect. We get the RNA sequence, and then our translate function processes it to produce the final amino acid sequence uh, for insulin. And we have just simulated the entire information flow from gene to protein. And that is a wrap for today. We have demystified translation by representing the genetic code in a Python dictionary, and then building a function to convert RNA into protein. And you now have the tools to simulate the entire central dogma of molecular biology. So, what's next? Now that we can generate protein sequences, a very common task is to compare them. So, in the next video, we will dive into the world of sequence alignment, where we will learn how to find similarities and differences between two sequences. And as always, the code will soon be on GitHub. And if you have questions, please pop them in the comments below. And don't forget to like and subscribe. I will see you in the next lesson. Thank you.